Hi guys, I wanted to go through some information about cells to help you out for your final half quiz of our cell unit. So as you prepare for this cell unit, I want you to be sure that you refer to Chapter 4 in your text and refer to all of the modules and credentials that are on Canvas. But hopefully this video will sort of give you a few more details, cover a few more ideas. You might also notice on my YouTube channel I have some other videos about parts of the cell. Those are for my honors bio class, but they would not be bad things for you to watch if you just feel like you have more interest in the organelles and what's happening in a cell. But those videos probably go into a little more detail than you might need to know. So let's start by talking about cells themselves. So, organisms are living things, and they're either single cells, which you've done a lot of thinking about, especially in your book projects. Things like prokaryotes and protists, they're just a single cell that does something. Or they're multicellular, such as plants, animals, and fungi. And so when we take all of the organisms on Earth that are made of cells or are cells, we're going to start off by just dividing them into two basic categories. And you probably remember learning about these in class. We have prokaryotic cells and eukaryotic cells. Prokaryotic cells are going to be bacteria and archaea. You probably know mostly about bacteria, and the key thing about prokaryotes is that they're not going to have a nucleus. So pro means before, karyo is kernel. So prokaryotic is before it had a kernel inside, before cells had a nucleus. So bacteria and archaea do not have a nucleus inside. And then the other category is going to be eukaryotic, a true kernel, eukaryotic. It's going to have protists, plants, fungi, and animals. These things are all going to have nuclei inside of them. So I sort of alluded to it a minute ago in the idea that prokaryotes are before eukaryotes. Prokaryotes are older than eukaryotic cells. They appeared about 3.5 billion years ago, and we only saw eukaryotes about 2.1 billion years ago. Both of those seem really far back, but remember there that we're talking about a difference of over 1 billion years between the first prokaryotes and the first eukaryotes. Prokaryotic cells, because they arise so much earlier, are smaller than eukaryotic cells and much simpler in structure. When we get to eukaryotic cells, that's where we talked about organelles. So only eukaryotic cells have organelles by the specific definition, which is that organelles are membrane-enclosed structures that perform specific functions. And we can think about this idea that when a eukaryotic cell gets a little bit bigger than a prokaryotic cell, instead of having an efficiency apartment where your bed, toilet, sink, refrigerator are all in one room, as you get to a bigger and bigger house, your specific rooms have specific functions. You have a kitchen for storing your food. You have a bedroom for sleeping in. The most important organelle that we're going to think about in a eukaryotic cell is the nucleus, primarily because that defines what a eukaryotic cell is. But the nucleus is also going to be the house of all of the eukaryotic cell's DNA. Now, the DNA is the material that tells a cell what it is and what it's going to do. And so it makes sense that you're going to put that DNA in a protected place, somewhere where it can be safe from any insults that might come across the cell. The nucleus, interestingly, is surrounded by a double membrane, and that's going to play into providing more protection for that material inside of it. So let's look at some of these different kinds of cells. This, I hope you recognize, is a prokaryotic cell, and you can tell because it's very simple, and in fact it does not have a nucleus. So the key things I want you to look for in a prokaryotic cell are that all cells, prokaryotic and eukaryotic, are going to have a cell membrane or a plasma membrane. That's going to be something that surrounds the cell. A prokaryotic cell is also going to have a nucleoid region, which is just a place where DNA is found inside of the cell. So this purple block is just DNA. And we call the region where that DNA is found a nucleoid. This is not like a eukaryotic cell. It's not bound or contained by anything. It's just free-floating. So we're going to have a plasma membrane, a DNA in all prokaryotic cells. We're also going to have ribosomes in all prokaryotic cells. And eukaryotic cells, ribosomes are going to be complexes that allow us to take nucleic acids and turn them into proteins. And since proteins do most of the work of the cell, it's very important that cells are able to make proteins. And this doesn't have it labeled, but the other required feature of both prokaryotes and eukaryotes is the cytoplasm, which is the liquid interior of the cell. So as long as the cell has a plasma membrane, DNA, ribosomes, and cytoplasm, it has enough to function. We'll have a few more adaptations in a prokaryotic cell that we can talk about. A cell wall is going to add a little more structure to the outside of the cell. That's going to help protect it from the outside environment. A prokaryotic flagellum, which is different than a eukaryotic flagellum, we'll talk about in a little bit. That allows the cell some motility so it can move about. And the pili are sort of appendages that stick out of the cell and allow it to attach to things. So it helps these bacteria sort of attach to a host, attach to one another, and sort of control their interactions with the environment. So we'll contrast this very simple cell with a more complex eukaryotic cell. So all eukaryotic cells are fundamentally similar to one another, and we already alluded to in the prokaryotic cell, everything else in the cell that's not a nucleus is the cytoplasm. So the cytoplasm has various organelles floating in it, suspended in a liquid that we call cytosol. So the cytoplasm is made up of organelles and cytosol. The cytosol is just the liquid inside. We have two kinds of eukaryotic cells. We have animal cells and plant cells. All of you are animals. The cells inside of your body are animal cells. Plant cells, on the other hand, are going to have chloroplasts that let them turn light into chemical energy. Consequently, chloroplasts make plant cells green, which is why plants are green. Plant cells are also going to have protective cell walls. Similar, although not identical, to those that we saw in prokaryotes. And these protective cell walls will help support the shape of the cell. So here's our idealized animal cell, and you can see the nucleus is prominent. Inside the nucleus is the DNA. Coming away from the nucleus, we're going to have the endoplasmic reticulum right near it. And we'll talk about all of these in a minute, but you have a rough endoplasmic reticulum and a smooth endoplasmic reticulum. You'll have a Golgi apparatus. It's going to look like a stack of pancakes or something. We'll also identify the mitochondria. You can tell a mitochondria because you'll see it's double membrane, and the inner membranes have all of these wrinkly shapes in them. The centrioles and the cytoskeleton are part of the skeleton of the cell. So these are going to provide support and help hold things together. We have our ribosomes again to make proteins, and we have a lysosome, which is going to help the cell digest stuff that it doesn't need. So you can see already the animal cell is much more complicated than that prokaryotic cell we looked at a minute ago. Everything is ordered, it's in its place, and it gets some benefit by having things in the appropriate places and knowing where they are. We can compare that to a plant cell, and by and large, the plant cell is going to look the same. Nucleus, endoplasmic reticulum, Golgi, mitochondria. We're going to add the chloroplast. And so you can see the chloroplast is green, and it has these stacks of membrane sacs. That's going to help the chloroplast carry out their function, which is to turn light energy into food energy. And on a plant cell, we're also going to add this cell wall. So you notice that the outside of the cell looks more rigid, is less round. That's because the cell wall is providing some more structure. Plant cells will also have a large central vacuole. So this is just a large membrane sac that can hold water for the plant. And if you think about it, a plant can't get up and walk to water. So it makes sense that evolutionarily it's adapted ways to sort of store materials for later. So let's go on a tour of all these organelles. Let's talk about them in a little more depth. You should definitely be sure that you can identify the organelles on a picture of the cell. Make sure that you can look for some of those features and then know what their functions are. So we'll start at the outside. We'll start with the plasma membrane. So the plasma membrane, or the cell membrane, is made up of lipids and proteins. And you may remember from our last unit that the lipids we're going to talk about are what we call phospholipids. So they're going to have two fatty acids and a phosphate. The fatty acids will be hydrophobic. The phosphate will be hydrophilic. And therefore, these phospholipids are going to form a two-layer membrane that we call the phospholipid bilayer. And if you think about that, the reason it's important that one part is hydrophilic and one part is hydrophobic is that the hydrophobic parts have to associate with one another, And the hydrophilic parts are going to associate with the water that's both outside of the cell and inside of the cell and create a nice sort of barrier to keep things from going in and out of the cell. Now, our next unit will talk in more depth about how this barrier works, but you should be able to recognize that some proteins can be hydrophobic and some proteins can be hydrophilic, and therefore proteins can be found on the outside of the bilayer, the inside of the bilayer, or going right through the bilayer, which we call an integral protein. We call the membrane a fluid mosaic model because it is fluid. It's not a static structure that's sitting there. It's sort of bobbing along like balls on top of a pool. Everything is moving around. There's a lot of flexibility. And so you have an opportunity for lots of things to interact at the cell membrane. So going from that cell membrane, let's go all the way into the middle now and talk about the nucleus. So the nucleus is going to be the part of the cell that directs the cell's action. And it's going to direct that action using genes in the nucleus. Genes are simply portions of nucleic acid that have a specific set of instructions. So we will talk about this later in the year, but a gene is a specific instruction or a specific recipe to make a protein. Since proteins do most of the work of the cell, when the nucleus stores those genes are ultimately what's driving the direction of the cell. Here is a picture of a nucleus, and as I mentioned before, the nucleus is a double membrane structure. You can call this the nuclear envelope or the nuclear membrane. That membrane is going to look just like the cell membrane, it's just in a different part of the cell. In fact, every organelle is going to be surrounded by a lipid bilayer membrane. Inside of this, you're going to have DNA in the form of chromatin, which is just DNA that's been wrapped up around some proteins to give it structure. Think about spaghetti being wrapped up on a fork. We're going to have a region called the nucleolus. The nucleolus is the region where ribosomes are being made inside of the nucleus. And then things such as RNA, messenger RNAs, or ribosomes are going to exit the nucleus through these nuclear pores. So there are holes in the nucleus that allow things to leave the cell. So importantly, think about the nucleus as being a place where the DNA is stored and protected. RNA copies of that DNA are made and pushed out, and ribosomes are also made in the nucleus and pushed out. There's an example of DNA being packed up with proteins into chromatin, and you can see it's just like spaghetti being wrapped around a fork. And then when those messenger RNAs, which are nucleic acids, leave the nucleus, they will go to a ribosome to be made into a protein. So ribosomes, like I said, are made in the nucleolus, and they are assembled in the cytoplasm. And in the cytoplasm, they basically read a piece of nucleic acid and turn it into a protein. We will spend an entire unit talking about how this process works. So for right now, all you need to know is that the ribosome is able to turn nucleic acids into proteins. Therefore, they have a pretty important role in the cell because proteins do everything in a cell. Ribosomes can make these proteins in two places, either in the fluid of the cytoplasm, so a free-floating ribosome, or if they are attached to the outside of an organelle called the endoplasmic reticulum. So when we say the rough endoplasmic reticulum, it is rough because it's coated with ribosomes, and those ribosomes are making proteins that are going into the endoplasmic reticulum. So here are free-floating ribosomes. This is a transmission electron micrograph. And you can also see ribosomes that are attached to a piece of the endoplasmic reticulum right here. So as we start talking about the endoplasmic reticulum, this is part of the endomembrane system. The endomembrane system is a system who manufactures and distributes products throughout the cell. And it's going to do this by shuttling proteins from place to place, either through their membranes or through membrane segments between them. So you notice that the ER was right around the nucleus. The ER is one of the main manufacturing facilities of the cell. It can produce an enormous variety of molecules. It's connected directly to that nuclear envelope, and it has both the smooth and the rough ER. We already know that the rough ER is that ER that's coated with ribosomes. The smooth ER, by contrast, is not going to have ribosomes on it. So here's our rough ER coated with ribosomes, our smooth ER without ribosomes. And the difference in functionalities are that the rough ER has ribosomes, so it's going to produce membrane proteins, and proteins that are going to be secreted by the cell. So when they go into the rough ER, or when they go into the rough NER membrane, they're going to be dispatched to other parts of the cell by vesicles. These are tiny little membrane sacs of membrane that can butt off of the ER and go to, say, the Golgi apparatus. You can see here's a ribosome. It makes a protein inside of the rough ER. When it gets to the end of the rough ER, it buds off into a little ball. That ball is a transport vesicle that can move elsewhere in the cell. The smooth ER, like I said, doesn't have surface ribosomes. So instead of primarily functioning to produce proteins, it's actually going to produce lipids and steroids. And in your liver cells, you actually have a large amount of smooth ER because the smooth ER will help detoxify circulating drugs throughout the body. So the Golgi apparatus was that stack of pancakes. And that stack of pancakes is actually the next step of the endomembrane system. So it's going to work in partnership with the ER. It's going to receive those materials that come from the ER, those vesicles, refine them, modify them, store them, and then distribute them throughout the cell. And so you may have talked about the Golgi apparatus as being sort of a FedEx facility or a shipping facility that decides on where things go and moves them there. So the Golgi in these stacks that we call cisterna, you have a receiving end, so a vesicle would come in to the receiving end. Things would move through these stacks, and as they move through these stacks, they get modified and changed and prepared for doing other things. And the other side of the Golgi apparatus, the shipping side, would help you move things out to somewhere else in the cell. So these transport vesicles could go to the plasma membrane and push it out of the cell. They could go to other portions of the cell. One of the other places it could go, if a vesicle does not go to the membrane, it could go to a lysosome, which is another membrane-bound sac that is actually full of digestive enzymes. So sometimes the cell makes things that it doesn't want anymore or wants to destroy. If this lysosome fuses with a vesicle full of protein, it can hydrolyze or break down proteins, polysaccharides, fats, and nucleic acids. So think of this as like a stomach. It's full of digestive enzymes, and when it interacts with a vesicle full of stuff, it's going to break everything down that's inside of it. So if we have a vesicle or a vacuole full of food, the lysosome could engage with it, and then everything inside will be broken down. The cell can actually put a vesicle around organelles that are damaged and fuse that with the lysosome and just break down, say, a mitochondria it doesn't need anymore, and then use those parts to build something new. So lysosome is a membrane sac full of digestive enzymes. Lysosomes can destroy bacteria, break down organelles. They actually are used to help sculpt structures during embryonic development. So your hand, at one point in development, was just a paddle, and the lysosomes helped sculpt tissues and remove the cells that were between your fingers. Vacuoles are just large sacs of membrane that bud from the ER, Golgi, or plasma membrane. Vacuoles and vesicles are quite similar structurally. Vacuoles just tend to be larger vesicles that can store stuff. We're not going to worry too much about contractile vacuoles, but some organisms need a vacuole that can collect a lot of water and pump it out to make sure that that cell doesn't have too much water. Here's an example of it here. Don't worry about that too much. Just know that a vacuole is a big membrane sac that can hold something. Plant cells have a large central vacuole, and we mentioned that is a unique feature to plant cells. And so you can see that the plant cell is kind of squished around, and most of the volume of the cell is taken up by a central vacuole. So this is a view of the entire endomembrane system, and you should be thinking about this as a pathway. That DNA comes from the nucleus, at the rough ER can make proteins. Those proteins can move through the rough ER until they bud off in a vesicle. Those vesicles will then move to the Golgi apparatus, and those proteins can move through the Golgi. And at the Golgi, they have some various places they can go. They can go to a vacuole and be stored for later. They can go to a lysosome and be digested and start over. or they can be pushed to the cell membrane, and when this vesicle fuses with the membrane, everything inside is going to be released to the outside. So there are three sort of places you can end up: outside of the cell, in a vacuole, or digested by a lysosome. Alright, so that was the endomembrane system. Now the chloroplasts and mitochondria are going to be separate from the endomembrane system. They are likely, based on the endosymbiont theory, were prokaryotic cells that were endosymbiosed, so basically eaten by a eukaryotic cell, and they mutually benefit one another. The eukaryotic cell gets the energy from them. The predecessor, the prokaryotic cell, got protection from the outside world. The two organelles are cellular power stations, chloroplasts and mitochondria. All cells, animal and plant, have mitochondria. Mitochondria is going to allow us to turn sugar into ATP. Only plant cells have chloroplasts. They're going to allow us to turn sunlight and carbon dioxide into sugar. Most of the living world, even including us, runs on the energy provided by photosynthesis. Everything we eat ultimately comes from a plant, which ultimately comes from the light from the sun. The sun is going to be utilized as energy to turn carbon dioxide into sugar and other molecules. Chloroplasts are only in plants and algae and do this process of photosynthesis. So here is a chloroplast. Features you should recognize is that it's a double membrane structure, and inside of the double membrane you have these membranous sacs that stack up that provides a lot of membrane surface area for the chemistry that needs to occur. For right now, you don't need to know a lot of detail about the inside of the cell, but you should be able to recognize what a chloroplast looks like as a double membrane thing with stacks of membranes inside. Know that this is green because it absorbs energy from the sun and uses that energy to make sugar. The opposite idea of a chloroplast in the mitochondria. Mitochondria allows us to do cellular respiration. All eukaryotic cells, including plants, have them, and they produce ATP from food. And one way to think about it is produce ATP from sugar. So again, this is a double membrane structure, and you should be able to recognize this by the fact that it has all of these wavy membranes inside of the outer membrane. And this provides a lot of surface area to the cell. And when we talk about how the mitochondria works, you'll see that this surface area actually allows the chemistry that needs to occur inside of this organelle to occur. And so you should be able to recognize this by its crista, by its invaginated membrane inside, and know that this is what you guys like to say, the powerhouse of the cell. It is the place that's going to turn food into energy that the cell can use. Mitochondria and chloroplasts actually have their own DNA, and this leads back to that idea that they were free-living prokaryotes that were taken up by our eukaryotic predecessors. The last thing to talk about is the cytoskeleton. So the cytoskeleton are fibers that are all inside of the cell. The cell would otherwise be a sack of liquid, except it has these structures that are just like a skeleton in your body that help hold the cell together, help the cell move. Provides mechanical support and help it maintain its shape. The three types you should know are microtubules. Microtubules are the largest. They're straight and hollow tubes that can be used by organelles and chromosomes to move around on. So think about it like a train track that different structures can grab onto and move. If you remember the video that we watched at the beginning of the unit about the cell, we saw that guy that was sort of walking along. It was walking along a microtubule. Intermediate filaments and microfilaments, or sometimes for microfilaments I'll say actin filaments, are thinner. And they also have the same functions as the microtubules, but they do them in different ways. All of them provide anchorage and reinforcement for many types of organelles. Just know that the cytoskeleton is a way of supporting the cell itself. Here are pictures of microtubules. You can see these yellow microtubules are spread out all over the place. The blue is actually actin, or microfilaments, that's giving structure in other parts of the cell. They can help a cell move around. So this is an amoeba. Actin will help it reach out and then pull itself up. There are two major modal appendages that are based on the cytoskeleton, the flagella and the cilia. And these are those structures that you might know about that beat to help a thing swim. They are pretty much identical structures. Flagella tend to be longer and there are fewer per cell. Think of a sperm cell. Cilia are more numerous and slightly shorter. Think about on the inside of your lungs, there are little cilia that help move all of the mucus out of your body. They have the same basic architecture and do things. So here's a sperm cell with a nice flagellum off of it. And here's a protist that uses these tiny cilia to swim, or cilia inside of your respiratory tract that help move things out of your body. Know that these are made of microtubules and many other proteins. About 250 proteins make up this structure, which is very different than the prokaryotic flagellum, which has the same function but a different structure. So that completes our tour of all of the organelles. If you kind of know the basic details that I just talked about for all of the organelles, you should be fine. Make sure that you're reviewing Chapter 4 and using the study guide for this quiz this week. And please feel free to reach out if you have any questions.